All right, so turgor, plant and bacteria have a rigid cell wall outside of the cell membrane. The cell wall, it prevents excessive swelling and bursting. And then turgor pressure is the outward pressure of the contents of the cell. So we can see in the cell on the left, right, the water is going into our vacuole, which is this part right here. But what's happening is all of the cell contents are pushing against the cell wall this way, and that's what turgor pressure is. Okay, as the plant wilts, the cell membrane is going to pull away from the cell wall, and this is known as plasmolysis. So if you guys remember doing the red onion cell part of the diffusion lab, we put that red onion cell in both fresh water and salt water. So in fresh water, we kind of had cells that looked like this, right? Our cell was nice and plump. But then as you put that cell in salt water, we could see that the cell membrane kind of pulled away from the cell wall. And that's what it kind of looked like on the right-hand side. All right, facilitated diffusion. So facilitated diffusion is a special kind of diffusion, and it needs the help of carrier proteins. So these carrier proteins are located within the cell membrane, and what they do is they can help bring larger molecules into and out of the cell. So you can kind of imagine them as like little doors that help bigger things come into and out of the cell. The molecules are still going to move from high to low concentration. And since we're still moving from high to low, it also doesn't require energy. So we're still looking at a form of passive transport because it does not require energy. Okay. So facilitated diffusion uses carrier proteins, molecules still moving from high to low, passive transport. All right, now we're going to talk about active transport. So active transport is the opposite of passive transport. So in this case, molecules are going to move from an area of low concentration to an area of high concentration. So when this happens, we call this going against the gradient. So we're going from low to high. Right? So we can kind of think about this as like rolling a big boulder up a hill. Takes lots of energy when going from low all the way up the hill to high. This doesn't occur passively. So what this means is that the cell is going to use energy in the form of ATP, and it's going to use what's called transport proteins that are kind of going to act as pumps to pump these molecules through the membrane, going from low concentration to high concentration. All right, so endocytosis. If we take a look at this word, we can kind of break it up into two parts. Endo, going in or inside, and then cytosis, talking about a cell. So when we're talking about endocytosis, a cell is going to engulf particles or fluid that cannot pass via protein transport. And what it's going to do is, if we take a look at this picture right here, we can see that our cell membrane is forming, this is called a vesicle, this area right here. So it's kind of forming around what it wants to bring into the cell, and it brings it in. So engulf means kind of like just swallow. So it's kind of swallowing whatever it wants to bring into the cell using the plasma membrane, and then it will suck it into the cell. Okay, so we're talking about two different types of endocytosis here. We have one where we're taking in particles of food, and then the other one would be liquids. So phagocytosis would be the process of taking in large food particles. And then penocytosis would be engulfing liquids. And both of these are going to be active transport. And remember, active transport means uses energy in the form of ATP. All right, last slide. So exocytosis. Can kind of guess this is going to be the opposite of endocytosis. So exo, like exit. Right? So in this process, we are going to move materials to the outside. So we want to get rid of things. So what's going to happen is our cell is going to form a vesicle around what it wants to get rid of. And then this vesicle will kind of merge with our cell membrane. And then eventually it's going to open up and then let all of these particles outside of our cell. Okay, when this happens, since we kind of have this vesicle that's merging with the cell membrane, this is going to increase the size of our membrane because we're adding more membrane to it. And this is the main way that we are going to secrete molecules. Okay, one other thing that I want to add is exocytosis is also a form of active transport. Remember, this means uses energy.